Thank you very much. It's really an honor to be invited here to speak tonight and to tell you about the work that we're doing at Agios to really try to understand the role that IDH mutations play in the development of cancer for the ultimate goal of providing therapies to patients. And the beginning of my talk is a lot of what Matt went through, so I'm going to go through this pretty quickly. But just to remind you, IDH is a metabolic enzyme, and it functions in the normal wild-type setting, to convert isocitrate to alpha-ketoglutarate, which Matt just went through. Back in 2008, mutations were discovered in IDH at this locus, the R132 locus in the IDH1 enzyme, and in the R172K analogous locus for IDH2. And as Matt pointed out, when people first identified this, they looked to see if IDH could convert isocitrate to alpha-ketoglutarate, but it could not do that, and so they thought that this was a loss-of-function mutation. And Matt showed you this slide again, and this is just, and I'll just repeat that when we did the experiments, and this was done by Yen, to actually see if IDH was able to convert isocitrate to alpha-ketoglutarate, only the wild-type enzymes were able to do that, and they used NADPH, this cofactor, as a readout of the activity of the enzyme. But at Agios, our company is focused on developing developing drugs against targets of metabolic enzymes. And the reason it was founded in this way is to think about new ways to target cancer. And maybe there was a possibility that IDH, because this mutation is always found over and over again, it's not what you would expect to be a tumor suppressor, as Matt pointed out. It just doesn't make genetic sense why a tumor would select to maintain this mutation. And so we did the exact same experiments at Agios that I showed you with Yen, measuring NADPH. And the wild-type enzyme makes NADPH, and that made perfect sense. And again, when we did the same experiment with the mutant enzyme, we saw that NADPH started to be generated, but then it was quickly consumed. And every time we put in the substrate, isocitrate, we would see this cycle again. NADPH would start to build up, and then it would quickly be consumed. And this was suggesting that perhaps there was another activity that this enzyme had, as Matt talked about previously. And he showed you, as we were looking in the biochemistry reactions, we also express these mutant, either the wild type or the mutant protein in cells, and look to see what is metabolically different about these two types of cells. And to do that, we did an unbiased metabolic profiling, and Matt showed you this exact same slide where we compared the mutant cells to the wild type cells, and these three metabolites stuck out like a sore thumb, and as when we went in to find the identification, lo and behold, it was 2-hydroxyglutarate. And that's really exciting, but again, we still weren't really sure what 2-HG was doing. And we went back and did the experiment to show, and I'm not going to go through this for the interest of time, but exactly what Matt pointed out is that the enzyme isn't converting isocitrate to alpha-ketoglutarate. It's actually running in the reverse reaction and taking alpha-ketoglutarate to produce 2-hydroxyglutarate. And sorry, this died. Oh, and in the process, producing 2-HG and consuming NADPH, and that's exactly what we see here by the mutant enzyme activity. So this was all really exciting. We had biochemistry evidence that this metabolite was being formed, and we had cell systems that we engineered that were making this metabolite, but that doesn't prove that it's important in cancer. And so to do that, we collaborated with Linda Liao, who's a neurosurgeon at UCLA. I came from UCLA, so I knew her. I called her up. I said, what do you have in terms of tumors that we can look at? And she provided us a set of low-grade gliomas that were blinded to us on their genetic status. And we ran these over the mass spec, and what we found was that there was 100% correlation of all the tumors that had an IDH mutation had millimolar concentrations of 2-hydroxyglutarate compared to glioma patients whose tumors did not have this mutation. And that really speaks a lot to saying perhaps this molecule is playing a role in the pathology of the disease. At the same time, we were also looking at IDH mutations IDH mutations were being described in leukemia. And so we also looked at blood samples from an AML patient that has an IDH mutation or a leukemia patient who does not. And again, you can see that it's elevated in their blood if they harbor the mutation. And as Matt pointed out before, more work is now being done with great work here at MIT and MGH and UT Southwestern to really be able to use imaging modalities to look at this millimolar concentration of 2-HG so that you can really track the disease as it goes. And so we're really interested in this to use it as a potential surrogate for treatment effect, but also is it telling us that there could be clinical benefit if we reduce 2-HG. So Matt showed you this again. When I joined Agios, my role, and I have to attribute this to David Schenkein, who's our CEO. He came in and he's passionate about following science to actually understand how we develop drugs and push them into the clinic so we can really pick the right patients to treat. And my job at the company is to, okay, well, now you're making millimolars 2-HG, what does that do? And that's great, we can track it and we can pick patients by their mutation status, but I want to understand if I give them the drug and I lower their 2-HG, what am I doing to their tumor? How can I follow that clinically and know that we're going to have a beneficial effect at the end? And as Matt pointed out, IDH mutations are found in multiple different tumor types, both IDH1 and IDH2. So I'm going to switch gears, and Matt told you about the glioma, but I'm going to discuss the work we've been doing in leukemia, AML, which is acute myelogenous leukemia, and some of the preclinical work in the first part of the talk, and then in the second part, I will tell you about the results we have from our clinical trial data, first initial data. So this is a breakdown of the IDH mutations in leukemia specifically. So IDH1 mutations are found and IDH2 mutations are found. But the problem is that no cell lines that we use in the lab that exist that are from leukemia patients actually harbor an IDH mutation. So we were kind of stuck. What are we going to use to study the disease? And so to do that, we relied on a model system, a cell line. It's called TF1. It's just, oh, thank you. It's a cell line that was generated from a patient with an erythroid leukemia, and it grows in the tissue culture dishes, and we can do experiments in the lab. But what we did was we overexpressed either the IDH1 mutant protein or the IDH2 mutant protein in these cells. And what normally happens, these cells require a growth factor called GM-CSF in order to grow. But if you express IDH1 or IDH2, these cells can grow in the absence of that growth factor. So it suggests that the presence of that mutant enzyme is conferring some novel activity to these cells, allowing them to rapidly proliferate. And this correlates with millimolar concentrations of 2-HG. We know that's what the enzyme's making. So I'm going to go through the data, but I always like to start with the model because I'm a visual person, and then when I see the model up front, then I can follow the story behind a little bit easier. So this is what we think is happening, what 2-HG does in a cell. As Matt pointed out, there's a whole host of enzymes in your body that require alpha-ketoglutarate, which is the normal substrate for the normal IDH mutation, to activate these enzymes called DNA and histone demethylases. And what these enzymes normally do is they put methyl groups on the DNA and the histone, and it quiets the DNA. But what we find is that and when you have these enzymes, they regulate the expression of genes in the genome, and that can have an effect on the regulation and the gene expression and actually the cellular differentiation. So what we know is that the IDH mutation, when it's expressed, it produces boatloads of this 2-HG, and this comes down, and because it's so similar in structure to alpha-ketoglutarate, it actually competes with alpha-ketoglutarate for binding to these enzymes and functionally inactivates these enzymes. So they no longer can remove these methyl marks on the DNA and the histone. And what happens when that happens is that the chromatin is condensed, and the genes that are involved in these regions are not expressed, and your cell sits in a de-differentiated state or a more stem state. So our compound targets specifically the mutant enzyme, which is great for clinic because you're not going and targeting all your cells in your body. You're targeting cells that specifically express this mutant enzyme. It reduces the 2-HG, and then functionally reactivates these enzymes, allowing the methyl groups to come off the DNA and histones, and then you get normal differentiation, normal expression of your genome. So back to the work where that led up to this model. Again, we're focusing here in these studies on IDH2 enzyme. I showed you earlier that you get this growth factor independent growth with expression of IDH2 mutation. And we looked at the methylation pattern on the DNA in these cell line and compared it to the methylation pattern of primary patient samples that had the IDH mutation. And when you do a principal component analysis, it's just a statistical analysis of these samples and see how they cluster together. What we found, interestingly, is that TF1 mutant cells cluster very closely to the primary patient cells that have the IDH mutation. And that suggests that their pattern of methylation is similar enough that they may share similar biology. So now we're off to the races. We have a tool cell line that we can then begin to explore the biology and the consequence of having these enzymes. So the first thing we noticed, and I always joke around about this, but the first thing we noticed is that the cells, just their morphology was different. And why is it different? And what we normally, TF1 cells float in a dish, and they don't sit down on the plate. But when we express the mutant enzyme, and Kaiko Kuni, who is actually here, I think she is the one who first noticed this when she was trying to expand these cells so we could do some experiments. She kept coming up and saying, these cells keep sitting down on the plate. I don't know what's wrong with them, and it's just funny, and we didn't know, and they have a spindle shape. And so that actually is a clue to the biology, and that suggests actually that they're more primitive in differentiation, that they're more mesenchymal rather than epithelial. And to test that, we looked for a marker of mesenchymal cells, which is vimentin, and you can see that it's very highly expressed in the mutant cells versus the vector. We also now we had a clue that these cells were less differentiated, so we looked at cell surface markers to look at that. And CD34 and CD38 are both expressed on the cell surface of the control vector cells, but on the mutant cells, actually CD38 is not expressed as much anymore, and that's another clue that these cells are in a more primitive state. So because we're a pharmaceutical company, we are developing small molecule inhibitors against these targets. And to do that, we're lucky because we can test the biology by using small molecules, AGI-6780. We can treat these cells and see when we shut off the enzyme what happens to the cells. And that's exactly what we did. The first thing we looked at, I told you the DNA gets methylated, so we wanted to see, does the drug reverse the methylation? And that's exactly what we saw. If you compare the vector to the IDH2 mutant cells, you see a lot more methylated DNA. And if we treat with compound for either 7 or 28 days, we now see a reversal of the methylation. And about 2,300 of these genes, which were reversed, we looked at what do they have in common, what kinds of genes are they, what pathways do they belong to. And the top 5 scoring hits were actually genes that have been associated with acute leukemia, as well as in stem cells. So again, this is making you think maybe we are in the right track here of what's going on. We also looked at the histone methylation. I told you that's another protein that gets methylated. And in the mutant cells here, in this column without drug, you see a bright band, which suggests and proves that they're hypermethylated on multiple histones, and that's not found in the vector cells. When you treat with the compound and we titrate in the drug and lower the intracellular 2-HG, we can dose-dependently reverse this hypermethylation on the histones, just like we can reverse the DNA methylation. We can also reverse the expression of vimentin, which is the marker of the stem cells. So again, suggesting that we're changing the biology, perhaps from a more stem phenotype into a more differentiated cell by treating with the drug and lowering 2-HG. And this is a functional assay to show that we are differentiating these cells. I told you these are erythroleukemia cells. So erythroid cells express hemoglobin and turn bright red when you challenge them with a growth factor called EPO. And that's in the vector cells. When we do that, and these cells don't have the mutation and don't produce 2-HG, with EPO, they turn bright red. The mutant cells, without with millimolar concentrations of 2-HG, in the presence of EPO, they fail to change color. And so they're blocked from their differentiation cascade. But when we treat with the drug, we cause the cells to turn red. I joke that my kids could do this experiment. And the hemoglobin also comes on, and that correlates with the change in color. And KLF1, which is a transcription factor, which is a master regulator of erythroid development, also gets expressed in the presence of the drug. So again, we wanted to take it one step further. This is a model system. It's not a patient sample. So we collaborated with Stéphane Dubaton at Institut Gustave Roussy in Paris, and he provided us with primary human patient samples that we can look at in the tissue culture dish in the lab. And we looked to see if we treated with drug, first of all, do we lower 2-HG? We know they have elevated 2-HG, can we lower it? And that's just an example in these two graphs showing that indeed we do that. But we wanted to see, are we affecting the differentiation status of these cells? Because this is what we saw in our model systems. And this is a complicated slide, but the point I just want to show you here is that these are fact studies which just tell you how the state of the tumor is. So in the mutant patients in this middle and to the graph on the right, when you have no drug present, this patient has 75% tumor cells, and when you treat with drug, it goes down to 53%. Here in this panel, we saw a reduction from 53 to 28. But the really exciting part of this is this works, and that looks exciting, but also when you treat a wild-type patient, there's no effect on those tumor cells, which goes to show that your drug is specific for the mutation, it's specific for these types of patients, and so that this will provide better toxicity profiles for when we go into the clinic. You're not just blasting every cell in your body, you're really having a specific effect. We also looked at the expression of cell surface markers for differentiation, and the CD11b, CD15, and CD14 are what we looked for. And in the mutant patients, we can see upregulation of their expression, showing that these cells are differentiating, and that doesn't happen in the wild-type cells. And this is just a graphical representation. A tumor cell in a leukemia is called a blast. And so you see without the drug, they have a high percentage of blast cells. But when you titrate in the drug, the blast cells disappear. And the pathologists actually can look at the morphology of the cells under the microscope. And they see not only the blast disappearing, but they're actually seeing these cells differentiating into more mature cell types. So we now have two in vitro systems where we see that 2-HG blocks differentiation and the drug reverses it. We wanted to then test in an in vivo model for cancer. So we did an experiment with AG221, and I'm going to switch gears now from the tool compound to AG221, which is our clinical candidate that we're using now in the trial. It's a first-in-class inhibitor of the IDH2 mutations. And just like I showed you the data for 6780, this compound is very potent against the mutant enzyme. It's selective against off targets. It can induce differentiation in the TF1 cells. It can induce differentiation in the primary patient cells. And when we treat mice, we can see that we can lower their 2-HG dramatically by dosing with this compound. So we set up to do the experiment, and this experiment is we take a patient's primary bone marrow, is where the tumor resides, and we inject it into mice through a tail vein injection. And 40 days after we inject these animals, we can see engraftment of the human tumor as a measure of CD45 in the bone marrow, spleen, and peripheral blood. And when we see the tumor engraft in these mice, the 2-HG in their peripheral blood also increases. So this is a nice model system. And eventually, these mice will all succumb to leukemia at around day 80 because they now just have such a leukemic burden. So we can use this, treat with our compound, and see, can we affect their ability of survival, and can we reduce the 2-HG? And so this is the first experiment that we ran. We did 3 different doses of AG221, 5 mg, 15 mg, or 45 mg per kg. And then we compared the ability of this molecule to affect the tumor compared to Ara-C, which is what's used in the clinic. And in the mouse study, this is what's called a Kaplan-Meier curve, and it measures the survival. And it's really exciting. We found that AG221 promoted a beautiful dose-dependent survival advantage to these animals. And in fact, at the high-dose group, the mice survived well past, and this is at 130 days, they went well past 160 days. We decided to take the mice down that were remaining still alive at this time point so that we could look at the biology endpoints and see, are we inducing differentiation, and does that differentiation correlate with their survival advantage? And that's exactly what we see. In the CD, as a measure of differentiation, we looked at CD15, which is a marker, a cell surface marker of a differentiated cell, in not only the peripheral blood, but also in the bone marrow where the tumor was. And we see that we have a dose-dependent survival, and the animals that died early in the low-dose groups were actually mice that failed to express the differentiation marker. And we see a reduction in blasts. I'm being told I have 2 minutes, and I want to get to the exciting spot, which is the clinical data. So this was data that we presented at EHA, which in June of 2014, and this was an update to where we are in our phase 1 trial. Again, a phase 1 trial, the main goal of a phase 1 trial is to assess safety and tolerability of your drug. And so we do a dose escalation. We start with a low dose and we titrate up, and we look to see how patients do. And as they tolerate the compound, we can decide what doses we can then go in to do a broader phase study. This is a summary of the safety implications for the drug. So to date, as of June, we have had no severe problems in terms of safety for the molecule, so the patients were doing well. And this is the PKPD analysis. So we are able, even at the lowest dose that we started with, we got enough drug on board in these patients to completely suppress the 2-HG, which is really exciting because then hopefully we can see some biology. And actually, this is the data to the efficacy, where we were able to look at 35 patients. 10 of these were non-evaluable, but 25 of them were evaluable, and their overall response rate was 14 of the 25 patients had a clinical response, and 6 of these 14 had a complete remission of their disease. And so that's very exciting. When you look at this data as a waterfall plot and we map out the response based on their dose and their cohort, what we notice up front is that in all doses we had a nice clinical response, but as we dose higher, we start to bring that response in earlier. And so these are some things that we're following to track as we go out longer in our trial. The other part of this is that we're collecting samples all the way through this so that we can monitor the disease and see what are we actually doing to these cells. And just like we saw preclinically, we see that that we induce differentiation in these patients on the trial. So when we started the drug, before they got the drug, they are all full of blast cells, but by 15 days on the drug, we see the appearance of more mature monocytes, and by 28 days, we see fully mature granulocytes, and the reduction of the blast cells, which is the tumor cell. These are the blasts at the beginning, and by the end, they're gone, and their white blood cell counts have normalized. So that's very exciting. And so in conclusion, the molecule AG221 that we're using is safe and well tolerated in the clinic. We can inhibit 2-HG up to 100% in our patients. We do this, the molecule works by inducing differentiation, and that's very consistent with our preclinical data. And we had 14 of the 25 evaluable subjects achieved a response, and 8 of the 25 had a complete response or a complete response without full platelet recovery, which is exciting. These responses are durable. We not only have the IDH2 trial ongoing, we have an IDH1 mutant trial ongoing, that is tested AG120 against the IDH1 mutant in advanced hematologic and solid tumors, and that data is not public yet. And again, this is everybody at Agios has touched the IDH drug in some way. This is a group from our Founders Day, and I can thank specific people, but I think at the end of the day, we really have to thank the principal investigators, the institutions they work for, and more importantly, the patients who are brave enough to take these drugs for us, and hopefully we can provide benefit to them. Thank you.